Hello everyone, welcome to Botany class. The topic what we are going to learn in this video is Mendel's principles. We know that John Gregor Mendel, who can called as a father of genetics, he conducted hybridization experiments on garden pea. The scientific name of garden pea is Pisum sativa. But he conducted hybridization experiment from 1856 to 1863. So this period you can call it as Mendelian periods, and based on his experiments, because he conducted the many number of hybridization experiments like monohybrid cross, dihybrid cross and a trihybrid cross using 7 contrasting pair of characters in Pisum sativa. And based on his hybridization experiment, he proposed some important generalization. The generalization together you can call it as Mendel's principles or Mendel's law of inheritance. And this law used to understand the pattern of inheritance in organism. And what are the law proposed by John Gregor Mendel? So first one is law of dominance. Law of dominance is the first law proposed by John Gregor Mendel. And the second law is law of segregation. This is another law proposed by John Gregor Mendel. And third law, law of independent assortment. So these 3 laws together you can call it as Mendel's principles or Mendel's law of inheritance or principles of inheritance also. Whatever you are using, everything same only, right? But among these 3 laws, the first 2 laws, law of dominance and law of segregation, these 2 laws he proposed based on his monohybrid experiment. So Mendel he conducted one experiment to study the pattern of inheritance of one gene at a time. For that he conducted monohybrid experiment. So based on that experiment, he proposed 2 important laws. One is law of dominance and second one is law of segregation. And he conducted another one experiment. So that is a dihybrid class to study the inheritance of 2 genes at a time. And based on the dihybrid experiment, he proposed another one law. That law you can say law of independent assortment. Okay. So these 3 laws together we are calling as Mendel's principles or Mendel's law of inheritance, or simple term you can say principles of inheritance. And what is the use of this law now? But based on this law used to understand the pattern of inheritance in organism, right? And before getting into these particular laws, we can recall the monohybrid cross conducted by John Gregor Mendel. Okay, you see, for why he conducted monohybrid cross now? To study the pattern of inheritance of one gene at a time. For that he conducted monohybrid cross. The cross which involved actually, so when we are taking the parent organism, here he want to study the inheritance of only one gene at a time. For that he conducted monohybrid experiment, right? You see, for that he selected first true breeding or pure breeding parent plants. First he selected tall plant, another one is dark plant. So the genotype of this tall plant is, because I already told it's a pure breeding variety, so that it's a homozygous, purely homozygous dominant, capsty. And dwarf also is a pure breeding variety. The genotype of this dwarf plant is small t. This parent organism, okay, produces only one kind of gametes, okay, gamete with this capsty gametes. And this one also produces only one type of gametes, that is a small t. And he hybridized these 2 parent organisms, which produces F1 progeny, nothing but filial progeny 1. So this filial progeny is, it's a tall plant, but the genotype of this filial progeny is, it's a heterozygous dominant. So because in allelic pair, So one allele is caps T, another one allele is small t. Okay, it's a heterozygous tall plant. And these tall plants, again by selfing, he produced F2 generation, filial generation 2. And this F1 progeny produces two types of gametes or two kinds of gametes. One is gametes with caps T, another one is gametes with small t. If you put Punnett square, we will get four type of progenies. So the progeny, you see, here I am taking female plant, caps T and small t. Here I am taking gametes of male plant, caps T, small t. If you put Punnett square, we will get, there are four progeny we will get. So out of four, three progenies are, like you see, this one and this one, these ones are, they are tall, and only this one is dwarf. The phenotypic ratio is 3:1, and the genotypic ratio of monohybrid cross is 1:2, so 1:2:1. Okay. So this is a monohybrid cross. Based on this cross only, he proposed two important generalization or two important law. One is the law of dominance, another one is law of segregation. Okay. So first we will start with law of dominance. So this law of dominance he proposed based on the monohybrid cross observation, right? The first law is law of dominance, right? So this law also called as Mendel's first law. The law of dominance also called as, so Mendel's first law also. First law. And the law state that, the law of dominance state that characters are controlled by. Characters nothing but the trade or the future of the organism is controlled by. So discrete unit. The discrete unit he called as a factors. So each and every character of organism is controlled by the discrete units. The discrete unit he called as a factors. So now we are calling this factor as a genes. But Mendel he called that one as a factors. And another, the same law state that these factors always occurs in pairs. So each and every character of organism is encoded by or expressed by the pair of gene, not by a single gene. That is given in law of dominance. So you see, the characters are controlled by discrete units called factor. Now we are calling this factor as a gene. So each and every character of organism is expressed by only genes only. And the last state that another important point given in this law is factor always occurs in pairs. So each and every character or trait of organism is expressed by pair of factor or pair of gene, not by single factor or single gene. Okay, you see here the tallness, first point, the character is controlled by discrete unit we are calling as factor. Here monohybrid class, this is a true breeding tall pea plant. For the tall, this character is controlled by, expressed by this pair of factors, caps T and caps T. And factors occurs in pair, we are saying that. Okay, you see here, the tallness of this plant is expressed by, not by single gene, the pair of gene, caps T and caps T. Similarly, the dwarfness also controlled by small d, right? So that factors occurs always in pairs. And the third important statement given in this law of dominance is, if a dissimilar pair of factors, dissimilar pair of factors, so if present together, dissimilar pair of factors present together, so one pair dominates the other pair. The dominant one you can call it as, so the dominant one, which the one pair which dominate the other pair, so that the dominance and recessive will be there. Okay, you see here in this F1 progeny, the progeny, all the progenies are looking tall, not looking exactly, they are tall plants, And the gene or factor which control or responsible for this tallness of plant is caps T and small t. But here in this pair, these are dissimilar pair, not similar pair. Here you can see parent organism caps T, caps T. This is a similar one. And here you can see small t, small t. This is a similar one. But here you can see this is a dissimilar pair. But in this pair, but only caps T is expressing, but small t is not expressed. So one pair are the factories which dominate the other one. So that the caps T can call it as it's a dominant factor and small t can call it as recessive factor. Okay. So these are the important statement given in law of dominance. Are you clear with this? So the law of dominance states that actually this law of dominance also called as first law of inheritance, the Mendel's first law. So the law of dominance states that characters are controlled by discrete units called factors or genes. Factors always occurs in pairs. And if the dissimilar pair of factors, if it is present, so one member of the pair dominates the other one. So that in heterozygous condition, only one is expressed, other one is suppressed one. And also this law used to explain expression of only one parental trait in monohybrid cross. Only one parental trait may be expressed in F1 generation. And also the expression of both traits in the F2 generation. Okay, see here tall plant is there, dwarf is there. What F1 plant in heterozygous condition, only one parental trait is expressed here. Here we are taken two parental character. One is tall, another one is dwarf. But F1, which one is expressed? Only tall is expressed, but dwarf is not expressed. And also this law is used to explain expression of both traits in F2 generation, filial generation 2. Filial generation, you see, here this plant is tall, this one is tall, this one is tall, but this one is dwarf. But in F2 generation, both traits or characteristics of the parent are expressed here. Like some progeny looking tall, some progeny looking dwarf, right? But this law also explain the proportion of phenotype is 3:1. In monohybrid cross, always we will get if only single gene is inherited, the progeny phenotype how it look na? The ratio of the phenotypic ratio is 3:1. We will get in F2 generation, and genotypic ratio is 1:2:1. One is a homozygous dominant, two is heterozygous dominant, one is homozygous recessive. But this law of dominance is, the law is not universally applicable. Not universally applicable. I hope that you understood the law of dominance. So next we will start with, we will see law of segregation. The law of segregation also called as Mendel's second law of inheritance, right? So guess law of dominance is first law of inheritance, and law of segregation is second law of inheritance. So this law of segregation is proposed based on the monohybrid cross. Okay. The law of segregation states that, it's a very important law, it states that during gamete formation, either parent organism or their progeny, F1 or F2 progeny, during gamete formation. So we know that, so each and every character of organism is encoded by, expressed by pair of factor or pair of gene. But during gametic formation, the factor of fact present in parents. Here parents, I am saying that either the master parents or maybe F1 progeny or F2 progeny parents. So the factor of pair present in parents segregate from each other so that the gamete receive only one of the two factor. So that lies law segregation. Okay, see here in a parent organism you can see. So the factors are present in pairs, caps T. But during gamete formation, what will happen? They are segregating from each other, so that this gamete receiving only one of the factor, not both the factor. This is a homozygous condition. But heterozygous condition, you can see here F1 progeny, the capacity is small t. These factor pair, these are dissimilar factor pair, but they are present together. But when the factors are present together, in the individual organism, they do not get mixed or they do not blend up. But the both factors are associated together, but they segregate from each other during gametic formation, so that the gamete receive only one pair of these factors, right? You can see here capacity small t is there, so this gamete receive capacity and this gamete receives small t only, not both of the factor. So that is given in law of segregation. Are you clear with this? So I can explain again. During gametic formation, the factor of a character pair present in parents, or that is an individual parent organism, they do not get mixed or blended. So simply they associate together, but they segregate from each other during gametic formation. So that during gamete formation, the gamete receives only one of the two factor. Okay, whatever condition, homozygous condition or heterozygous condition, whatever may be, the gamete receive only one factor from that pair. So either dominant factor or recessive factor. But if it is homozygous parent, produces only similar gametes only. Okay, so here you see this is a homozygous parent, capacity, capacity. This one also homozygous parent, small t, small t. Here it produces only one kind of gamete only. So gamete with only capacity, and if it produces two gametes also, all are its similar kind of gametes only. Here also it produces only similar kind of gametes. But if parent is heterozygous, the heterozygous parent produces two kinds of gametes, and each having one allele with equal proportion. Okay, see here, I can explain here. The capacity small t is there. This is one parent organism. Here this parent is heterozygous parent, because the factors, the particular for characteristics, they are dissimilar pairs. During gametic formation, what will happen is, it produces two kinds of gametes. One is gamete with capacity, another gamete is gamete with small t. But all the gametes, they are having only one alleles. So one allele, so the gametes they segregate from each other or independently. This given in law of segregation. So this law of segregation also called as the second law of Mendel's second law, or second law of inheritance also. But this law is universally applicable. But the law of dominance is universally not applicable. But law of segregation is universally applicable. Universally applicable, it's applicable for all sexually reproducing organism. Okay, all sexually reproducing organism, right? So next one is Mendel's third law. The third law also called as law of independent assortment. I forgot to tell one point. The second law, Mendel's law of segregation, also called as Mendel's law of purity of gametes, right? You keep in mind. The second law of inheritance, also called as law of purity of gametes, right? The third law is the law of independent assortment. So Mendel proposed this third law based on his dihybrid cross. Okay, the first two laws are purely based on monohybrid cross, because that cross involve only one contrasting character. But the law of independent assortment is he proposed based on his dihybridization experiment. Here the cross which involve two characters. Okay, here cross involve two character. This law of independent assortment states that when more than one pair of factors are involved in a class. Okay, you see dihybrid class, how many factors are involved? Two factors are involved, or two characters are involved. Similarly, if you take trihybrid class, 3 characters are involved, and tetrahybrid, 4 characters. So like if the hybridization experiment, when more than one pair of characters are involved in that particular class, so a particular class, the segregation of one pair of trode is independent from the other pair of the trode. So that is given in law of independent assortment. Are you clear with this? So when more than one pair of factor are involved in a class, during gametic formation, segregation of one pair of trode or the one pair of character is independent to the other pair of the trode. That is given in Mendel law of independent assortment. Let us see what is exactly the law of independent assortment. Okay, you see the dihybrid class. Here the class involve 2 different characters. Here parent 1 Mendel bot is selected for this hybridization is so the seed color and seed shape. So the first parent is which produces seed which are yellow color and round shaped. These are purely both characters are homozygous and dominant one. And the second parent is which produces seeds which are green color and a wrinkled shaped one. In that he made class, he conducted dihybridization class. Okay, see here these are one pair of factor, this is another pair of factor. Here also, so the color is one of the factor and shape of the seed is another factor, right? During gametic formation, okay, see the Y segregation is independent from the R segregation. Similarly, R segregation is independent from the Y segregation because they segregate separately, not depends on the each other. So that the gamete receiving, like monohybrid, in out of 2 pairs, gamete receiving only 1. See, caps Y, caps A is there, the gamete receive only 1 Y. Here also caps R is there, here it receiving only caps R. Similarly, small y, small r and small r also. This F1 hybrid, here the factors of pair, they are present in heterozygous condition. The dissimilar pairs are there, like caps Y, small y, caps R and small r. But this F1 progeny produces yellow color seeds and which are round shaped also, right? Round shaped one. So these are heterozygous condition but dominant character is expressed. So now you see the heterozygous condition during gamete formation. So one pair of trode segregation is independent from the other pair. The Y segregation is independent, R segregation is independent. Both are not dependent. Okay, you see the Y that there is it produces the gametes with 50% of caps Y. Similarly, 50% of small y gametes. Equal proportion will get gametes. Similarly, R also produces 50% caps R and small r. So that what kind of gamete you will get? You see caps Y like this segregate and caps Y along with caps R. Or small y along with small r. Or caps R along with small y. So small r along with caps Y. Like that we will get 4 different type of gamete. Because one gamete segregation, so one para factor, their segregation is it's not depends on the another pair of the factor or another pair of the trode. This is given in law of independent assortment, right? Are you clear with this? I will explain one more time. If more than one pair of factor involved in a class, in this class 2 factors are involved or 2 pair of characters are involved. Okay, so that's why we are calling as dihybrid class. So during gametic formation, segregation of one pair of trode is independent to the other pair of the trode, right? You see, this is a heterozygous garden pea. Here, the 2 characters are involved here. One is a seed color, another one is seed shape. During gamete formation, so the factor responsible for seed color segregate independently from the factor responsible for seed shape. Okay, so that you will get the 4 different type of gametes. Okay, like caps Y, caps R, small y, small r, and caps R, small y, small r, caps Y. Okay, so that we are getting 4 combination. Okay, see, if it is not the factors are one factor segregation is depends on other, then how we will get? We will get only 2 chances are there. Either we will get like this. So small y, caps Y, small caps R, or small y, small r. This chance only we will get. Or either we will get like this, or we will get caps Y, small r, and small y, caps R. Either we will get gametes like this or like this only. But the factor segregation is independent. That's why we are getting the 4 type of or 4 kinds of gamete here. So this one is not possible. Okay, this one is not possible here. I hope that you understood the Mendel principles, law of independent assortment, so the law of dominance, law of segregation or law of purity of gametes, and Mendel third law, law of independent assortment, right? I hope that you understood the concept. So up to this end of this video. The next video will continue further topic. Thank you.